Okay. You should have received also the message.
Yeah, we should press OK. Okay. So I would just thank our speakers, which are Naama Brenner and Elisa Franco, for this third talk of this series of online seminars. And then we'll leave the floor to Alessandro Rizzo, which will introduce the speakers and make the presentation for them.
Thank you very much, Mattia, and thank you to everyone that joined us. And I hope that in the next few minutes, we're gonna have more people connected. I'm honored to chair this session and this joint talk and conversation with Naama Brenner and Elisa Franco. So as many of you know, maybe you attended already many other seminars in the SICC series. This is a series of seminars that substitute actually our annual meeting that has been in person. Of course, due to the pandemic emergency, we started last year with a series of talks, but we thought to have guests in pair, so we can then stimulate a conversation afterwards between the two speakers and of course with the audience. Today, today is dedicated to conversations on biological systems. Our two speakers today are Elisa Franco and Naama Brenner. Naama, who is going to be the first speaker, she's a professor at the Technion Israel Institute of Technology in Haifa, at the Department of Chemical Engineering and Laboratory of Network Biology, and she's the head of the Cell and Population Biophysics Theory Group. Her group studies cell and population biophysics, and they're interested in variability, dynamics, and adaptation at the interface between cell and population levels of organization. They are part of the Network Biology Research Group at the Lori Loki Interdisciplinary Center for Life Science and Engineering, and they aim at bringing together researchers from different Technion departments to join forces and investigate fundamental problems in biology by quantitative approaches from different disciplines. I'm going to do a proper and longer introduction to Elisa, so people don't forget the introduction. Meanwhile, I just mentioned for the moment that she's an associate professor at University of California, Los Angeles, and then I will go with a proper introduction afterwards. Just to recall the rule of the game, actually each speaker has more or less about 40 minutes for a talk, where of course you can ask quick questions. I think there will be no problem if someone wants to interrupt, so raise a hand and ask question, or maybe we can get also quick question at the end of the first seminar, and then we will leave the floor for another 40 minutes to Elisa for another talk, and then we have scheduled 20 minutes for the proper debate. So I invite everyone to ask quick questions maybe after the talk or during the talks, but then reserve the, let's say, fundamental and longer questions that deserve more attention for the final debate that will last more or less 20 minutes. So I think it's time to start. The first talk by Naama is entitled Sloppy Control in Bacterial Growth Homeostasis, and it's with great pleasure that I leave the floor to Naama. Thank you. I can't hear. Okay.
Okay, can you see my screen and can you hear me?
Of course, yes.
Great. All right, so first of all, let me thank the organizers for inviting me to this nice series of seminars. I would like to talk about bacterial growth homeostasis today. And I start with a very nice picture that I came across. So I guess what I want to say with this picture is that people have been fascinated by the growth of bacteria, their growth and division for really many, many years. And this is a hand-drawn picture out of Henrici's book from about almost 100 years ago, that he was looking at how the bacteria grow and then how they divide and how their number, he was interested in their number, how the rate of growth of the number of cells, the rate of growth of biomass. And I think he's considered one of the fathers of microbiology, but already at that time, people were very interested in this process and to understand it. It's a very fundamental process. These days we use, of course, microscopy to look at bacteria, and the development of time-lapse microscopy enabled us to follow this and see how bacteria actually grow and divide in real time, and to make quantitative measurements out of this. But this, you can easily appreciate that such measurements under a constant field of view have the drawback that they quickly saturate. So you can watch this for a few generations, but then the environment is saturated. And so more recently, people have started using microfluidic devices to follow single cells for a much longer time. So I'm having some problem with my cursor, so movies are not going to work, I guess. Okay, anyway, these recent technological advances basically trap single cells and follow them for a very, very long time. And for example, this device, now known as the mother machine, traps a cell, and then the daughter cells are separated from it, and then they're washed out by this channel over here. And what this amounts to is the ability to track actually a single cell at the bottom of the trap for many, many generations, many more than you were able to do with simple time lapse. And because the growth and division and homeostasis that has to do with it is something that happens a long time, this is a very powerful enhancement of technique, and it enables us really to see this process over long times. So here's what it looks like. This is a measurement of cell size, actually cell length, because the cell width is more or less determined by this trap here. And what we measure here is the cell length as a function of time. This is a very long time. This is basically as long as it gets. Maybe the world record in such traces, and there are about 250 cycles of growth and division, and the cell cycle goes up and down. It grows and divides. You can see that it's a fluctuating process, but you can also appreciate that it doesn't diverge. It remains stable. So this is a really concrete way of seeing homeostasis at the single cell level under these trapping devices. So if you zoom into one of these cycles, then what you find is that cell size grows very smoothly, continuously throughout the cycle, very well approximated by an exponential growth. So if I start from an xn at cycle n, and I grow for a time tau n with a rate alpha n, this describes the data very well. And then I divide abruptly, almost instantaneously, by a fraction fn, which is close to half, to start the next cycle. And essentially all the variability in this trace that we saw at the top can be described by this coarse graining, by assigning just a few parameters to every cycle. And all of the variability is encoded in the variability of these parameters. So, for example, the fraction changes from cycle to cycle, the time changes, the rate changes, there are distributions of these quantities, and that describes the fluctuations in this trace. And you can then write, you can then describe it very compactly as a point or a stochastic process that is basically a multiplicative process. I multiply the initial size by a factor of e to the alpha tau, or e to the phi n, the logarithmic fold growth, and then I divide it by the fraction of division. So I multiply, divide, and every one of these discrete parameters in the process are basically stochastic, they're variable, and this is a really faithful description of the data. So if I use this initial size, for example, I'm now taking this trace and I'm only plotting the initial size as a function of generation for the 250 generations, you can see that it fluctuates, but it's quite stable. And from a mathematical point of view, if I had a point process like this that I would approximately multiply by 2, then approximately divide by half, and so on, and these things were independent random variables, then they would not be stable. Then fluctuations would accumulate, and the process would be much less stable than we see here. And a really nice way to see this in the data is if you collect together the distribution of these factors, the growth factor and the division factor, and you just shuffle them around without maintaining the order, this is what you get. So in this gray curve on top of the data, what I did is I just draw at random one of these factors and one of these factors without keeping the order in time. And this is a very clear way to illustrate the fact that correlations are involved in this, in homeostasis, in keeping this process stable and not very largely fluctuating. So this is a logarithmic scale, and if I shuffle the order of the generations, I get maybe 10 or 20 times larger or 10 or 20 times smaller bacteria, and that really doesn't happen in nature. And so a lot of people have looked at this process in recent years. It's approximately 10 years since these kind of microfluidic devices were introduced. And there are many ways to explain this, but one simple way is to say, to notice that the fold change, the growth factor in this process is negatively correlated with the initial size. So if a cell has happened to be born a little bit larger than average, it will grow by a fold factor a little bit smaller. And this is enough to explain the distribution of these cell sizes that are relatively narrowly distributed. And like I said, this has been at the center of a lot of interest in the past 10 years, and a lot of models have been studied and suggested, mainly about cell size homeostasis, but also about growth rate homeostasis. And on this background, what I would like to focus on today is a little bit more systems-wide or systems-level aspect of homeostasis, because in a growing population, it's not only cell size or generation time, and not separately, but everything together is held at the homeostasis balanced growth. So exponential growth rate, and if you track protein content, also proteins, all of these variables and the division fractions, everything is really very stable, statistically stable. So what you see here is the first and second half of the traces that I showed before. And the distributions are quite broad on one hand, but they repeat quite nicely between the second and first halves of these traces. And so we really have a lot of variables, and we have cell size, and people have looked at the difference in cell size. You can look at actually the fold growth, like I said before. You can look at every one of the time or rate variables separately. And all of them are coupled to one another. These are not independent variables. And in a lot of studies that people have done, they're all correlated, they're all coupled to one another. And really the main question that I want to address today is, how do we know, I mean, first of all, phenomenologically, are some variables more regulated than others, okay? Or is it all of them more or less the same? Because the distributions are not that different. The standard deviation over mean is not hugely different, but can I find a way to say whether some variables the cell cares about more than others and regulates them? And some of the motivation for this comes from models that we have worked on that shows that in an interacting system, if you have dynamic coupling, it's enough that one variable is strongly regulated, and all the other ones, by virtue of their coupling, their interaction to it, will also seem as regulated variables. They will seem stable statistically over time. So how do we know? So our goal here is to try to look at the data and say, how do we know what the cell cares about? And the way we think about this is inspired by engineering theories or systems theories in engineering that highlight the sensitivity of system variables to different perturbations. So in systems engineering theory, you can define robustness of a variable, and that is sometimes mathematically defined as a logarithmic derivative, for example. And basically what you're looking at is the response or the variability under very small changes in the environment. So the idea is to try to extract how sensitive the different variables are to the environment. The problem is, of course, that it's very difficult to create experimentally a range of environments that we can control and that is not too largely changing. And so how do we design such an experiment? So here's the idea that we had. We look at the traps of these microfluidic devices, and each one of them produces this trace of cell sizes from which we derive all the other variables that I talked about. And all of them live in the same experimental system. So the experimenter is controlling the medium and the temperature, and some systems are really very highly controlled in incubators, etc. And they're all supposed to be exactly the same, identical conditions. But in fact, what we see if we take two such large traces as we looked at before, so these are again the initial sizes, the coarse grain variables as a function of time over 250 generations, we see the fluctuations in one trace and the other one also fluctuates, but they are removed with one with respect to the other. So each one of them has a different distribution, and this difference does not average out over 250 generations, which is a very long time in bacterial terms. So what we thought of doing is using, so there's obviously some kind of variability in this setup, in this experiment. So we decided to use this variability and try to understand how it affects the different variables. So we're basically taking an uncontrolled variability, we're using it as a distribution of environments, and we're going to study how the different variables respond to that. And so here's the hypothesis, if you like. We want to say that maybe a robust variable is one that has a very robust set point. And if that is different, if that would have been different between traps, then the system would compensate for this difference and hold the set point to be exactly the same. So this is a stiffly controlled variable. On the other hand, if I see a variable that is drifting between the different channels, which are, like I said, almost exactly the same, right? They're very, very similar. But if I see a variable that's very drifting between them, then this is a sloppy control. This is a variable that the system does not care about, quote unquote, and does not invest effort into holding it at a specific value. Okay, and we want to quantify this. So maybe this is a good point to ask if there are any questions. Okay, so I hope this is clear so far. And I realize, I mean, a priori, this is not, you can argue about this hypothesis, but what I would like to do is take it further, and then we'll see what we learn from it. And so this is the hypothesis intuitively, and what I would like to do is to quantify it. And to quantify this, I use the law of total variance, and I separate the set point or the time average from temporal fluctuation. So suppose I have a variable that I measure across time. It has a temporal average, it has fluctuations around the average. By definition, these fluctuations have zero mean and some variance. And now if I take the total ensemble of all traces pulled together, that has some variance, and I can decompose the variance to two contributions. One of them comes from the set point being variable between traps, and the other one comes from temporal fluctuations over time. Okay, and this is additive because they are uncoupled. And this is an exact equation. So the variables are different in their units, and so I want to get a dimensionless unit. So I divide by the total variance here, and I get two fractions that I call gamma trap and gamma noise. They add up to 1. One of them tells me how much the set point is variable, and the other one tells me what is the contribution of noise around the set points. Okay, so I have two numbers. And if you think about these traces as if they were dynamical system realizations, then this gamma trap quantity that we define here is exactly an ergodicity breaking parameter that was defined in the literature. And this quantifies for us how much different trajectories mix over time, and how much, in some sense, averages over time are the same as averages over the trajectory. So, but in our case, this relates to two different traps. It's the same mathematical quantity, but it has a somewhat different meaning. So this is the quantity I'm going to be interested in. And looking back at these two illustrations, this one has gamma trap equals to 0, and this one has gamma trap equal to 0.6. And with my hypothesis, I will say that this is a stiff variable that's held very tightly, and this one is a floppy variable that has a high gamma trap. Okay, so now we're ready to apply this to our data. And here's what we get. So what I'm showing here, I have many variables. I'll explain, I'll remind them that there are a lot of letters here. I'll remind what they are. And here on the y-axis, I have this gamma trap that is, as I said, a number between 0 and 1. So I have alpha, which is the exponential rate of growth of biomass. And that is really, that's a really high contribution from set point variability. It's more than 40% of this total variance of this parameter comes from modulations in the set points. The same with the cycle time. So these are the two time rate variables, and they're quite floppy, 40 and 30% variable. But maybe a little bit surprisingly, their product, which is phi is the logarithmic fold growth over the cycle, that has a much smaller gamma trap, which means that the set points of these variables are much more tightly controlled. The fraction at division is again goes up to 20% almost, but there's a variable here that, if you can see, it has a gamma trap of almost 0. So the gray horizontal bar here is the noise level. This is a statistical estimate of the finite size sampling effect. And this variable here has modulations of the set points that are smaller than even the finite size effect. And what is this variable? It's actually the product of these two things in my point process, the growth term and the division term. And actually this variable tells me by how much the size changes throughout the entire cycle. Okay, so this is really a strongly controlled variable. Cell size itself is quite sloppy at more than 30% contribution of set point variability. But interestingly, the difference, and the same for the final, I'm not showing it here, but this is initial size and final size are quite sloppy. But interestingly, their difference is much more robust. So something interesting is happening here with the combinations of variables. So if I summarize what I've learned here, actually not what I've learned, but what are the questions that this opens? This opens up two important questions now. So one of them is really these broad ranges, these broad, large sloppiness of variables. Where does it come from? Why are these slightly different traps that are so highly controlled with the best technology that we have? Why are they so different from, what's so different between them from one another? And the second is about the combination of variables. So like I said, cell initial and final sizes are sloppy, but the difference is less. Growth rate and time are sloppy, but their product is much, much less. How does this come about? Can we describe, at least phenomenologically, what underlies these really large changes in the sloppiness of different variables? Okay. All right, so first I'll start with the first question. And I ask, where does this come from? So why does the set point of different variables change potentially so much between two traps? And I can think of two types of reasons or two types of sources for this variability. One is internal and one is external. So it could be the epigenetic and genetic variables, the intrinsic cellular state variables inside every single cell. Because in every trap, of course, there's a different cell. And we know that cells in general are not identical, really. So that's one source. And the other source is that despite the fact that we're trying to control the trap environment, they are still different enough to induce these differences between traps. The problem is that the mother machine that I showed you before cannot distinguish between these two sources because they are different both in the internal and in the external factors. So what we do to solve this problem is we go to a different kind of microfluidic machine, which is called a sisters machine, very recently developed in the lab of Hannah Salman in Pittsburgh. And what they did is they took two traps and put them together in a V-shaped microfluidic trap. And what this does is that it holds two cells at the bottom of the trap in a very, very nearby environment. Basically, the medium is mixed between these two cells. And then we can we can basically decouple these two sources of noise because the two cells at the bottom of this trap, on one hand, they share the environment very closely. The environment is almost identical, whatever it is. But on the other hand, if they're not sister cells, then they have different lineages, they have different internal properties, and all of the internal variables are distinct. And the way that this decouples is we can use the law of total variance again, and the gamma trap that we had before decomposes or splits into two terms. I'm not going to go into the technical details, but this is quite simple. It splits into a lineage factor and an environmental factor. And now I can do the same analysis on pairs of cells that I grow in this sisters machine and see what is the contribution of each one of these sources that I talked about before. And here are the results. And so to summarize first, the result is that the environment is the driver, the main driver of these differences. And what you're seeing here is, again, this set of variables, exponential growth rate, division time, cell size, etc. This y-axis is, again, the gamma trap. The whole bar is the gamma trap, the contribution of set point variance. But now we have two colors that tells us how this splits between lineage and environment. And as you can see, the blue part, which is the lineage part, is really small. It's approximately 5% for almost all of the variables. Whereas those that are really very sloppy are sloppy because they're sensitive to the environment. So most of the contribution here comes from the microenvironment in the different traps that are slightly different from one another. Okay. Now, as I said before, one might worry that the distribution of environments and the way we look at this robustness question is not very well controlled. So this graph relative to the previous graph gives us at least some confidence about the robustness of our results, of the experimental results in the analysis, because this is a completely different machine, it's a completely different device, it's a different experiment. But you see that without the splitting, if you only look at the bar heights, this is a very similar picture to the one that we had before from the mother machine. So this gives us some confidence that there are both the numbers and more importantly, the relative numbers between the different variables. So this implies that really something is going on here with what the cell cares about and what it keeps regulated. Okay, so now I'm going to the next. Okay, so sloppiness comes mostly from sensitivity to the environment. And really, let me just add one more sentence. If you think about exponential growth rate, for example, at the single cell level, this is not that surprising that it's so sloppy, because metabolism is complex and it's very sensitive to small changes in the medium. And as cells are dividing, they're also secreting metabolites to the medium, and every microenvironment is really a little bit different than the next. Okay, so what I would like to address next is the second question relating to how what's going on here with the combinations. Why is alpha tau so much more robust than every one of its factors separately? And to try to understand this, what I'm plotting here in this graph, I'm plotting the set points of growth rate, alpha, exponential growth rate, as a function of the set point of generation time. Now, remember, every point here is an average over time of many, many generations. So I have many different traps here. Everyone holds a stable equilibrium, a stable homeostasis, but it holds it around a very different set point. Okay, so here it holds homeostatic growth around half an hour, and here about around 0.6. However, they're very highly correlated with one another. So how can we understand this correlation? Roughly speaking, E. coli bacteria divide in half, right? So on average over time, I expect divisions to be equal to half. So if that's the case, then the fold growth should be on average 2. And what this means is that the two factors that make up the fold growth, right? So e to the alpha times tau is the fold growth. That should lie approximately on the line of log 2, of tau times alpha equals to log 2. And this is exactly what we see here. So what this means is that the cell doesn't care what is alpha, it doesn't care what is tau, as long as they compensate. I have one lineage here and one lineage here that differ by significant fraction in their specific parameters, but they bother to keep the product on this line in order to have that is appropriate for the division in half. So that's one thing, but actually not all of them are on the log 2 line, and there are deviations here. So if I look at these deviations in the next step, at the next level, if I plot this here, I can think, so what happens if the cell really doesn't grow twofold over the cycle? So that can in principle be compensated if it also doesn't divide exactly in half. And this is what you're seeing here. There's a second level of compensation between the fold growth and the fold division. And the dynamic range of this correlation is quite small, actually, but it's still a very significant and high Pearson correlation coefficient relative to the ranges. So you can see that the fold growth times the fold division is kept very tightly around this value of 1. Okay, so this is another way of saying that what we said already before, that the total fold change in cell size over the entire variable is very close to 1, and this is the R variable. So here's the qualitative picture that explains this part of the results. I have 2 sloppy variables, the exponential growth rate and the division time. They co-vary with one another very tightly to ensure that alpha tau is approximately a fold growth of 2. And the deviations from this set point are compensated at the next level by the division fraction until I get a very strongly regulated variable, which is the total fold change over the cycle. Okay, and this is how the hierarchy of sloppiness is supported by these set point covariations over long time. So these are basically long time correlations, that extend over much longer time, many generation times. They're not over one single cell cycle. And here's a nice geometric way of looking at it. So now if I create this 3-dimensional space of growth and division variables, I have the exponential growth rate, the time, and the fraction. So what I see is that in this plane, the set points of the different lineages lie on a plane, on a manifold. It's actually a nonlinear manifold. If you look from the side view, you can see that they're quite held tightly on the manifold. But if you look from a front view, then you can see that they are pretty much scattered. I mean, they're quite variable inside this manifold. So really, there is no set point for bacterial growth and division. There's a set manifold in some sense. And things are fine, and homeostasis holds, and fluctuations don't accumulate, as long as each lineage remains at some point on this manifold. Okay, so the directions in the manifold are the sloppy directions, and the directions outside the manifold are the stiff ones. Okay, I'm going to skip this. It's a 3-dimensional movie, but it's not important and it's not working. But the main thing I want to say next is that all of this structure that we've been talking about until now is completely blind to cell size, right? Because you can double size and divide by half and go on doing this in a correlated and constrained way, but you can do this at any cell size. So what happens to cell size? What happens with that? So I go back to this plot, this first results plot that we had for the different variables, and I remind you that cell size was quite a sloppy variable, right? And I'm trying to figure out now whether this set points co-vary with other variables. And so the first thing I notice is that, of course, cell size variables co-vary among themselves as they have to, as they must. Because if I'm holding one lineage at an average size of 2 and another lineage at an average size of 3 at the beginning of the cycle, then at the end of the cycle, they have to be twice that of the initial on average. So of course they co-vary with one another, but actually if I try to reproduce now the correlation between fold growth and initial size, I see quite a surprising thing, and that is that these are okay, let me go back to the first slide that I showed. These are the negative correlations on a cycle-by-cycle basis, and we reproduce them on a cycle-by-cycle basis. Here they are plotted as contour plots, the same data, but when you average over time and you look at the set points, actually the set points do not co-vary. So what we find with cell size is that cell size variables they obey the obvious relations that they have to, but they do not couple to the growth and division variables over long time scales. Okay, so they couple only over short time scales. Okay, we can do this, we can go on and on, show more. I think I'm going to skip this because I want to leave some time for discussion or questions. And I'm going to summarize the results as follows. So the first conclusion is that growth and division homeostasis can be maintained for many generations around distinct cell set points, of size, growth rate, and generation time. The cell doesn't care about that if I want to use the first formulation that I used. The second thing is that trap microenvironment is something that the cell is very sensitive to. But maybe surprisingly, internal lineage properties are not so strongly affecting this variability. Most importantly, we did find that some variables are more directly regulated than others. Some are sloppy, some are held stiff, and the sloppy variables can co-vary with one another to compensate and form robust combinations, and therefore defining a set manifold on which all of these lineages can grow, can sit anywhere on this manifold. And cell size coupled to growth and division only on short time single cycle timescales, and not as a covariation of set points over long timescales. So just two afterthoughts before I finish. If you're familiar with the work of Jim Setna on sloppy systems, then obviously you can notice the influence of this on our thinking here. And what Setna has been working on for a few years now already is to understand parameter sensitivity in different systems. And what they find is that usually systems behavior or systems emergent properties are sensitive only to a small number of combinations of variables. And here I use the same display that they use in their sloppy systems display and show you the range of cell size relative to the range of this ratio R that we talked about. And this is much more, a very large difference in the ranges. Okay, I'm going to skip the next thing and go to my acknowledgement slide to enable just a few questions if there are any. So this is collaboration with Hannah Salman and students from Pittsburgh. And thank you for my funding sources, and thanks a lot for everyone here for listening. And if there's any short questions, I'd be happy to answer them.
Thank you, Naama. So I asked the audience for let's say quick questions, quick curiosities to fix ideas. Okay, maybe I have one, but from a very, let's say, general point of view, how does the experimental framework look like? So how do you do these measurements? So how do you measure cell size, cell growth? Which kind of instruments you're using?
This is all under a microscope, I think.
Okay, microscope.
Yeah, this is all under a microscope. So actually, what Hannah does in the lab is this is all under a. I mean, did I have a picture? I think I did. Yeah. So this is what the device looks like. You can also tag proteins, and then you have fluorescence, but you can also picture this with a bright field microscope. Usually we do both. We have both images. And so this is everything is under a microscope.
Okay, thank you very much. Any other question from the audience? I think there's Mario Di Bernardo raised his hand.
Yes, Naama, thank you for a very, very nice talk. I'm sorry, I'm in an airport, so I didn't get the beginning. Maybe this question addresses something that you basically said already. I just wanted to know, when you say sloppy control, etc. So I know the mother machine, and I guess this is all carried out in microfluidics, right? What is the control variable here?
So, okay, so this is kind of turning the problem around, right? The cell is presumably controlling things in order to stay stable. And what we are doing is we're trying to develop an empirical way to actually infer what is regulated in one.
I see. Okay. Are you planning in the future to also manipulate some other variables and see how external changes to the environment
Oh,
affect?
yes, definitely. This is a great question. And so this is the slide that I skipped actually. Is you can ask what is the functional role of such a manifold? And what we think is that the functional role will show itself when we go to dynamic experiments and see how cells change between environments.
Exactly.
Exactly.
Okay, thank you so much.
Any other questions?
Thank you, Mario. Any other question from the audience? I don't see raised hands. So for the moment, I thank very much Naama for this very interesting talk. And I passed the introduction of Elisa Franco. Thank you for being with us. Elisa is an associate professor in mechanical engineering and bioengineering at the Samueli School of Engineering at UCLA, University of California, Los Angeles. Elisa works at the convergence of structural biology, dynamics, and controls using specialized biomolecular frameworks. She's the recipient of the NSF Career Award in 2015, and she's an Hellman Family Foundation Fellow for the years 2013-2014. The title of her talk is Ultrasensitive Components Enable Adaptation in Molecular Feedback System. Elisa, the floor is yours. You can share your screen.
Thank you. All right, do you see my screen now?
Yes.
Yes, okay, all right. Thanks for the introduction and for the invitation. So I think my talk builds nicely on what Naama just talked about, because here we're looking at actually ideas on how cells may be able to be engineered for controlling levels of specific components inside them. So we tie back to the concept of homeostasis and adaptation that you just discussed. So I will go fast on the introduction, but adaptation is everywhere in nature. It's present at the level of organs, entire organisms, and even collections of organisms. And adaptation, what does it mean? Well, basically, imagine you all have experienced this when you have bright light that is hitting your retinas. What happens is that the pupil contracts, right? And what does this mean? It's trying to control how much light gets to your optical nerves and finally goes to the brain. So there's a certain optimal light intensity, an optimal area, and your body adapts the area of the pupil to make sure that you don't exceed or go below that particular optimal light intensity. So the goal is to maintain a constant light intensity that goes to the brain while you're adapting the pupil area. Okay, so this is a feedback process, and automation engineers are very familiar with this concept. You all probably have cars, and you have a car cruise control system when you want to maintain a particular speed in spite of irregularity of the road, wind, rain, slope changes. What happens is that your car the controller, the computer in your car will try to maintain the constant speeds by adapting the fuel to the engine in spite of disturbances. And so the ingredients, automation engineers have figured out what are the ingredients of a feedback control system now for probably 100 years, right? So the idea is that you have a process that may be subject to disturbances. We measure the output of that process, compare it to a reference, and based on the error, there is a machine, a controller that will adapt the inputs to the process so that the disturbances are eliminated. The effects of the disturbances are minimized, okay? So in the case of the car example, you're adapting fuel. In the case of the eye, going back to the example of adaptation in your body, you have adaptation of the area of the pupil. And then, there's one important concept in control theory is the idea of proportional and integral feedback. This is basic linear systems theory example. Is probably what any undergraduate, the only thing an undergraduate will remember after taking a feedback control class, the difference between a proportional and integral controller. So in the case of the car example, suppose you have a slope disturbance that hits at some point during your trip, while you're trying to go from 40 miles per hour to 50 miles per hour. That's a dashed line. So if you have no control, what happens? No feedback. What happens is that your car will be, the speed of the car will be drastically affected by the slope. But if you implement control that is simply proportional to the error, you can make up for the disturbance. I mean, you don't have so much perturbation, and you can improve how well you respond to that perturbation by increasing that constant kappa p, that proportional constant. But a key to actually maintaining the speed you want exactly is the presence of integral control, which means that you are looking at the history of your error and taking an integral of that. So I will mention integral control later on in my talk. So this slide is really just to kind of bring back memories to those of you who may not remember these concepts. All right. Now, let's move to the actual topic of this talk. The question that people have been asking for a while now in the era of genetic engineering and synthetic biology, how do we build molecular feedback controllers to control gene expression? So, there's the beautiful movies that you've seen before that Naama was showing where cells are dividing. Well, while they're doing that, they're producing a lot of proteins, okay? And protein production starts by reading the genome, the DNA of those cells. The genes get transcribed into mRNA, so the information is copied into this intermediate molecule. And then from mRNA, there is translation into proteins. The proteins are what makes up the cell. Imagine a cell is about 60% water, and the remaining 30% is largely protein. Okay, those are the molecules that are key to building the cell and to performing metabolism, and all the other functions that it has. So here in this context, we ask the question: how do we keep constant protein level when we have disturbances that could be, perturbations in the levels of, environmental variables like nutrients, temperature, and so on, but also internal, meaning that, there could be fluctuations in the components that produce RNA or that translate proteins for a variety of reasons. And so how does the cell make sure that it tightly controls level of essential proteins? So that's a question for understanding how the cell works, but in the context of building genetic systems, gene networks, it's really an engineering question. How do we build robust protein expression? So let's now go back to the picture of a system, of an engineered system where, we, as an automation theorist would think about, which is we have a process that we want to control and we want to add to that a controller. Is there a framework to think about that? And so I'm going to discuss, the ideas that a former student in my group, who is also now a postdoctoral scholar, Cristian Cuba-Samaniego, so he developed these ideas throughout several years. And so the idea is, how do we build this controller? And it's very simple. So now we think about input-output maps of the process and of the controller, being agnostic about the specific details of the process. But imagine that at steady state, you can map the output of the controller versus the output of the process. And you say, well, let's suppose the process has this kind of map. It's a nice, smooth, monotonic map, which is not uncommon in biology. Like, what is suppose we can build a controller that has this kind of input-output map. It's also monotonic, but it has the reverse sort of shape. And as you can see, the intersection happens at one point. So the only admissible equilibrium is that in the blue circle. So if you can build a controller like this, and if that steep transition is what an engineer would call a reference, it falls at a particular level that we call a reference, then even if the process has perturbations that could happen because of internal or external disturbances, so that gray line could be shifted up and down by quite a bit or even change slope, but the intersection will still fall near that reference. And so you could say that based on the width of this transition from low to high, that could even give you a measure of how much error you have. So we can say that this, if we could build a system like this, it would give us quasi-integral behavior at steady state, because you would essentially, the equilibrium would return to the reference even after a transient perturbation, merely because of the shape of the controller input-output map. And so the error also would depend on the width of this transition between low to high, but you could also map it to the local gain, which is the slope of this map. And so a kind of curve like the orange one, we call it an ultrasensitive curve. And I just want to spend a minute defining that a bit more precisely. So suppose you have now this gray curve here. You can look at the input level that is necessary to achieve 10% of the maximal output response, and the input that takes you to the 90% response. And you look at the ratio between the two. Okay, so if this gray curve becomes steeper, of course, those 2 points start converging toward each other. And so the ratio between them tends to 1. So in an ideal situation where, this is the steepness is so high that this turns into a step function, then this coefficient will be 1. And so this is a way of measuring how ultrasensitive a curve is. Now, I would like to mention that this type of concept of ultrasensitivity in engineering is often called high gain, right? Because you can think of the slope of this curve as the gain. And this mechanism has been used in electrical circuits for a while. Think of a comparator that has a very high gain. Maybe the gain is unknown, and so in this sense the device may have variability. But when you put it in the context of a feedback loop, if this gain, all you need is that this gain is sufficiently large, and then you get an input-output map that is actually tightly regulatable by the ratio of the two resistances in the feedback loop. So you can actually, in this case, track input and output very precisely. All you need is that the gain is large. So, we're looking at a similar idea in biology. So now the nice thing about thinking in terms of ultrasensitivity and high gain is that there are a lot of chemical reactions. There are a lot of reaction motifs that give you an ultrasensitive response in biology. So for those of you who are in this field, you have probably heard of cooperativity. This means that when you have different proteins or different proteins or small molecules that need to be present in multiple copies to have a certain regulatory effect, then we have these Hill functions where we have the Hill coefficient n, right? And you probably have heard before of the fact that as this Hill coefficient goes up, the curve becomes steeper. Then there's the reaction of molecular sequestration, whereby you have two molecules that bind to each other and form a complex. And if this interaction is sufficiently fast, the input-output map also looks like a steep curve, and we will return to this later. Then there's zero-order reactions, where you have inputs that activate or deactivate a certain output, and by, if you pick the parameters right, you get a very steep response. And then finally, there's devices that operate as switches, and we will return to this later in the talk. An example is enzymes that do recombination of DNA and can switch on and off expression of a protein merely by inverting the direction of a promoter. So let's now focus a little bit more on the molecular sequestration aspect. I wanted to mention that there is a lot of work that has been published in the last few years arguing about how sequestration is actually essential to build integral feedback in biology. And our contribution to this picture was that of saying, well, let's take a step back. Maybe we don't really we need sequestration. It's not essential, but what is essential is that this device is ultrasensitive, that the controller is ultrasensitive. And so we proposed a motif, a reaction motif that achieves ultrasensitivity. And in this case, it does include a molecular sequestration step, but it's accompanied by a downstream reaction that switches on and off. So let's look at this. So the first stage is basically what we call a comparator. So we have two inputs, A and I, that produce intermediate molecules. And these intermediate molecules are in some way complementary. So they bind to each other with a certain rate constant gamma. And this has a buffering effect. So this works as a comparator. Then, depending on which molecule is more abundant, the one that kind of is not the rate limiting step for the sequestration, can either activate, so brings this inactive complex to an active form, or deactivate. So the second input drives the reverse reaction. And this is what we call the switch part of the motif. So if we look at the input-output maps, again, these are steady-state maps. What you see is that sequestration in isolation gives you this, these curves that are steep, but may not necessarily be very steep. The switch in isolation gives you a Michaelian, like a slow increase in the output, but the combination of them gives you a very steep increasing map in this particular case. But what is nice about this motif is that depending on which input you operate, so you can fix one input, in this case, the inhibitor. So if you fix it, that will determine the threshold here that blocks the increase of the curve. But if you block A and increase I, you get the opposite effect. So the idea is that you can use it both as an activator and as an inhibitor, depending on which input you fix and which one you vary. So this reaction network has a relatively simple model, only with 3 differential equations. And I don't want you to spend too much time on it, but let's focus for a minute on the sequestration reaction. I've mentioned that before as an important component of it. And the comparator in isolation, what it does, what we found is that it basically computes the difference between the two inputs. It calculates the error. So one way to show it is to do a change of variable and define these 2 dummy variables, W and Z. And this allows you to decompose the network into a part that is linear and a part that is a Riccati type equation. And in this case, it's easy to do a timescale separation. And in the limit, for the sequestration rate being really fast, you can demonstrate that either variable will converge to the difference of the 2 inputs, even if they are time varying. They may be time varying. So if you could take X as an output or Y as an output, depending on which input you fix. But in both cases, you get the difference between the inputs. And you get a map, like an output map that looks like this, like a steady state map that looks like the one on the bottom right. So this is the first stage, okay? Now, if we put everything together, the analysis you can do is you can show that the overall network is a positive system, that it is structurally stable. And by structurally, I mean that it's true for any choice of positive parameters. And then there's ways to derive equilibrium conditions, always assuming that there is sufficiently fast sequestration, so that you can decompose, map the input and the output of the controller and relate them through basically a constant term that depends on the inhibitor, on the threshold in this case, plus some delta element that is really measuring the width of this transition. And if you do the math, you come up with an ultrasensitivity condition that relates various parameters. And it looks, like a, complicated inequality. And, might say, well, okay, of course, there's going to be combinations that give you ultrasensitivity. But I think this shows, in a way, the strength of this mechanism, in that you're not requiring any particular variable to be pushed to the limit of being like extremely high or extremely low. Instead, you're making a requirement on a combination. And in this sense, it makes me think of what Naama was talking about earlier. Cell division appears to occur, in a very precise way, but you could have, sloppiness in different variables, but their combination gives you tight control. So in a way, this is a similar concept. So if you do, we did computational analysis of the input-output map. And so we figured out, which ones are the variables that affect the most the shape of the input-output map. It looks like the ones that are more important are the ones associated to the degradation, the dilution. Okay, so each variable here has a certain dilution constant. So if you dilute too fast, then you lose this ultrasensitivity property, all else being equal. And also the switching parameters, alpha C and beta C, the ones that control how fast the output activates and deactivates, those also need to be fast. But again, their combination matters more than the absolute value of each of them. The results of the computation are comparable to the analytical result. So next, this is going to be used as a controller, right? And you can ask, well, what happens if you connect this chemical reaction network to a downstream target? I apologize, the build is incorrect here, but suppose you take the controller, let's focus on the top right here. And now you use the output U to bind and unbind to a particular molecule downstream, which could be, for instance, a gene. So if you assume that this reaction is fast, then you can, decompose. You can have timescale separation, and you can approximate the effect of this binding and unbinding on the total amount of output that is available. And if you compute how the input-output map changes as a function of this downstream binding site, of course, the curve is going to change because you're essentially subtracting output available. So you can see how the maximal level goes down. But if you normalize this curve relative to the total level available of U, then there is actually low sensitivity in the sense that the shape remains the same. The ultrasensitivity is not affected by much. So you will still have that nice steep transition. So then let's look at closed loop performance. So let's put this controller to work with an example process. So the goal here is to keep a constant output, adapting the production speed of the particular process. So here we're looking just at a vanilla model of gene expression producing, a gene producing a protein. So first of all, we have a way of estimating the maximum gain analytically for this simple network, and then we can find bounds on the error by relating the maximum gain to the width of this transition area, but also to the gain, to the maximum gain. We have ways to relate the two. And then next, we can actually look at the kinetics. Okay, so we did a bunch of simulations, and these are only for illustrative purposes. But what happens, for instance, if you have a perturbation in the process? This is what we called adaptation early on. Suppose you have a perturbation. How does the closed loop system respond to that? And so here we're looking at perturbation of the production rate of the protein. From these kinetic response, we can see that the output eventually returns to the reference level. There's, of course, a transient, there's a damped oscillation, but the system returns back to the original reference level, which is the red line. What happened when we look at the input-output maps is that, as you can see, we have essentially moved the gray line because of the perturbation. But fortunately, the intersection between the gray line and the orange line, which is the controller, remains near the reference. So this is the principle we were trying to illustrate at the beginning. So I wanted to make a point here. As you can see, the kinetics are slow. Okay, so but here we're only making requirements about the steady state. And so it's important to remember that these curves, these ultrasensitive curves, are steady state map, not kinetic maps. So we looked at adaptation. What if we have a changing reference? Throughout these simulations, the reference was constant. So if you change the reference, the system is also able to adapt, to track the reference. What happens in the steady state map is that in this case, the controller map moves. So the orange line, as the reference was changed, is moving to the right. And in this case, the reference is really just a concentration, because if you remember at the beginning, I explained, well, what you do is you fix one of the two inputs, and that defines the threshold of the response curve. And that's the key of that controller that we are examining. So now let's go back for a minute to the question of comparing this controller to sequestration controllers that were examined by extensively in the literature in the past few years. So sequestration controller, and compared to ours, only requires this reaction to happen. Okay, two components inside the network, they bind to each other, form an inert complex, and this reaction kind of depletes the variable in excess. So as I highlighted earlier, so yes, this type of chemical reaction can actually compute the difference between the inputs. And so in that sense, you can build a curve that looks like basically an absolute value curve. And actually, if you push this parameter phi to be almost zero, you can increase this slope. Okay, so you can see it from this expression in the middle. If phi is very small, then basically this curve, this input-output map will become very steep. And so you approach a perfect integrator, a perfect step. But the problem with this controller is that when you do have this parameter phi, then it doesn't work very well. And this is what one could call a leaky molecular sequestration controller. So here you have a simulation that shows you how, here you have gene expression process that you're trying to control. Upstream of it, you have the controller. Now you look at the output of the process, and you would like it to align to this reference R. So if you have a very low degradation rate, Y will be very close to the reference. But as this degradation rate phi becomes larger, you end up with a lot of steady state error. The discrepancy between the output and the reference becomes large. In our case, so when we replaced the sequestration alone with the brink controller with the same amount of dilution, the controller is actually not affected the performance is not affected majorly by the fact that you have dilution. So here you see an example simulation with the same process, parameters all the same, except that we have this additional reaction of switching this is a component of this brink controller. So this is great. But of course, there's no free lunch. And an important assumption of this controller is that the total amount of output should be conserved. Okay, so that's actually an assumption that I didn't mention at the beginning, but it's key for this to work well. So in fact, if you actually include production and degradation of the output of the controller, the presence of these two reactions, if these production and degradation rates are too large, what happens is that the performance of the Bring controller becomes worse. So here we're looking at same simulations as earlier for the leaky molecular sequestration controller, where we're varying the dilution rate. And on the right, we are varying production and degradation of the output of the Bring controller. So, there's no free lunch. So we get an improvement in terms of resilience to the dilution, but we pay the price of having to tightly control the output total level, okay? And so this issue, so the fact that the sensitivity to the dilution might matter for systems where dilution is significant. So there's, for example, bacteria divide very fast, and so this dilution rate will be large. In mammalian cells, dilution happens much more slowly. So in that case, you could probably get away with using molecular sequestration alone. But in bacteria, it might be relevant to actually use a controller that is not sensitive to dilution. So when can this reaction work well? Well, suppose that naturally the cell conserves the output total amount. For instance, if it's a DNA binding site, total amount of DNA in a cell is conserved. So if you were able to design a controller that has this type of architecture and the total amount is conserved, then you're in good shape to use this architecture. And here is an example simulation where we implemented this Bring controller through transcription factors that are heterodimers. And if both are operating on the same promoter, which is basically the output U, so they can activate or deactivate the promoter to control production of a protein output, and then that protein output regulates one of the two species that is sequestering, then what happens is that you can create this high gain that I mentioned before, and it's quite robust to changes in parameters. So here you can see this simulation. We looked at systematic perturbations of all the parameters in the network and compared the input-output relationship in closed loop. This would be the dark points versus open loop. And you can see that in open loop, you have a non-linear response. So there's not perfect tracking, but in the closed loop, you have you have a linear relationship between the two. It's not a one-to-one linear relationship, it's rescaled, but it's linear. Here, the plot on the right here shows, how frequently in all these cases you get, ratio between Y and R that is, close to 1. It's a bit moved to the left because, of course, as I said, this is not a one-to-one scaling, but most of the ratios fall into a narrow area. Whereas for the open loop case, this light blue curve, we have a flatter distribution of ratios. And we also did stochastic simulations because, of course, in a cell, you sometimes end up with a low number of copies for each component, and our stochastic simulations show that this type of controller can work well in that case too. So now I want to dedicate the last few minutes to giving you the sense of how you we're trying to implement this controller actually using an on-off switch mechanism that relies on recombinases. And this is work that was led again by Christian Cuba in collaboration with Ron Weiss and the group of Domitilla Del Vecchio. So basically, going quickly, so recombinases are proteins that invert DNA sites, okay? And so we're looking at kind of the top part of the circuit here. So the recombinase would be this variable R sub A, and it induces a DNA binding site to switch from inactive to active. So once you have that switch, there is the ability to produce a certain reporter, okay? So I don't want to spend a lot of time explaining how recombinases work because it's actually complex. Just bear with me. It's a protein that will bind to a particular DNA site and cut the DNA and flip it. And so there's many types of recombinases. The ones that they're using are 5C31. They're serine integrases. And they work as dimers. So you need 2 copies of a recombinase to do the job. They perform inversion and excision, and it's an irreversible process. So because they're dimers, you can actually implement sequestration through a mutant that dimerizes to the recombinase and sequesters the catalytic domain. Okay, so that's how you can implement sequestration. And here is an example. These are example data. These are preliminary data. They are unpublished, but it shows you how. So in the x-axis of each of these heat maps, you have the level, level of recombinase plasmid, and in the y-axis, you have the level of recombinase mutant that was transfected. And the color of the heat map corresponds to how much green reporter is being expressed, okay? And there were different levels of total reporter transfected into the cells. And so the take home, like what you should take home here from these plots is that in all these curves, you can see how there's kind of like a linear separation between the light green and the dark green that is moved as, you know, you have more reporter being produced. But if you fix for any of these plots, so let's say the one in the middle. So if we fix a particular recombinase level, you know, as the mutant level is increased, so the more production of mutant is there, basically the higher the threshold for producing the output. So it becomes lighter green over here because there's a lot of sequestration happening. And there's a lot of dark green over here at the bottom because there is little sequestration. So the recombinase can do its job without a threshold. So finally, you know, you also need to have the bottom part of the controller. And that is, so you need to be able to reverse the operation of the recombinase. And we do that with the 5C31 recombinase directional factor fusion protein that will flip back the DNA to the position, to the configuration it had prior to being recombined. And we still have that mutant dimer that operates sequestration, and it can actually do it on both. So, so this is what happens when you have the recombinase mutant. We tested how it can, like, the reporter is now placed in the opposite position. So the complete system is now can be built because we have the, you know, sequestration reaction that happens on both recombinases, and we have the ability to switch in one direction and reverse the switch of the DNA. The total amount of DNA is conserved. So this is, you know, a good implementation of the controller. It satisfies that key assumption that you need to have a total amount of output, of controlled output, be tightly regulated. And so here are preliminary results where, you know, you have in open loop, when you don't have the full control of the system, basically the expression of the output is all over the place. But when you have closed loop, so you have both the switching back and forth, then you get a tighter regulation of the output. So here you have the distribution of output. Now, these are obviously preliminary data because we still have to tune individual parameters to figure out whether the controller, the performance of the controller is being is consistent with the model prediction, and we need to compare it also to maybe other types of controllers. But it's encouraging in the sense that it's at least we can get a quite dramatic difference between the distribution of output in open loop versus closed loop. So to summarize, I have described a method to decompose, to think about engineered gene expression, gene networks into how like a control theorist would think about it, like having a separate controller and a process, and you can, you can adapt the controller depending on the process you have. Many controllers would work. In our framework, we suggest that an ultrasensitive controller where this the transition of the ultrasensitive map, the transition point can be decided and can be mapped to the reference. That's a helpful way of thinking about the design, and also it allows to like clearly decompose, you know, like when you build a controller, you have two things you want to control, you know, you want to control the reference and you want to control the performance. In this sense, you know, if you can build something that has a settable reference, so you can set separately the transition curve and the gain of the reaction network, that's a good way to kind of completely separate the two processes of design of you know, controller design relative to the process you're given. And so we suggested one particular chemical reaction network that can achieve this, and it relies on sequestration to be able to tune the reference, to be able to tune the threshold, and it needs to be ultra-sensitive, and that's what allows you to tune the gain. And so in terms of outlook, we have been thinking about ultra-sensitive controllers, how could they work to control population dynamics, and so there's a recent paper in the American Control Conference. The leader here was Cindy Wren at Caltech. So she looked at how population dynamics could be, you know, while the level of individual cells could vary by quite a bit, but if each cell has an ultrasensitive controller, overall the population will be able to track a reference. And then there's another project in preparation where we are actually trying to use the idea of sequestration in the context of building neural networks using molecules. This idea is not completely new. There have been other studies that have linked sequestration-based networks to the ability to generate ReLU-like functions. But Christian and Andrew Morman were able to identify a set of biological implementations that work in mammalian cells. And so this is still in preparation. So with that, I want to thank Christian. I want to thank Caitlin. She's an undergraduate student in my group that helped with some of the mathematical derivations, and then collaborators that have you know, advised us over the years on this project. And with that, I conclude. And if you have any questions, I'm happy to answer.
Thanks to Elisa for this very interesting talk that puts control in a new perspective, at least for many of us, in an unusual perspective, with a new application. And I ask at first if there are questions from the audience on this talk before I open the debate or ask something myself. Mario.
Yes, Elisa, thank you so much for the talk, and apologies for the background noise. My question was, in terms of metabolic burden, I mean, it's a very smart idea to use recombinases, but in terms of metabolic burden, did you think about how it compares with the classical sequestration type of controllers?
Well, it depends how you implement it. If it should be the same if that output molecule if that's a DNA,
there shouldn't be
it shouldn't change much.
Okay, interesting. Thank you.
Thanks.
Thanks, Mario. Any other question from the audience? Okay, actually, I have a curiosity that might be very naive, but since you made reference to electrical engineering, and my curiosity is whenever we do control of electrical systems, electronic systems, we can do in some sense resort to hypothesis of impedance matching or separation between the blocks when we reason with block diagrams in control using, we know that there's an infinite impedance or zero impedance, so we can let either the voltage or the current pass and, I mean, govern this coupling between the units. Here I see that whenever what I realize of course, as a naive observer is that whenever there is a coupling, everything is totally changing. Yes. So how do you, I think it's tricky to manage these kinds of coupling. So I was just wondering how, if this is really a problem, and how you
It is.
manage it.
It is. It is. In fact, there's a lot of literature on this that was spearheaded by Domitilla Del Vecchio at MIT. She was one of the first people to actually pinpoint the problem that you're mentioning. But it's been a well-known problem in chemistry for a while. Like, it's hard to decouple things because to communicate, to transmit information, you need to have things that bind, okay? And as you have a binding event, it's unavoidable that something gets depleted, right? Because you're sequestering mass, and then that propagates, that perturbation propagates. So how to fix it? So there are ways to fix it by introducing reactions that have very distinct inputs and outputs. Oftentimes you assume there is a catalytic process, meaning that, you know, something gets transformed into something else, but, you know, you make assumptions of, oh, well, you got to have a large pool of a particular variable that gives you an infinite, you know, it's like having a reservoir of fuel or like infinite voltage, sort of, to make sure you have this, as you said, you know, matching of impedances, decoupling inputs and outputs. But another thing that I want to point out is that actually nature has, in the end, evolved compartments like electrical circuits, right? So you have the cell, as we for a long time we have thought about cells as just bags where everything is well mixed, but it's not true. So cells actually segregate components, probably because you need to have separation between reactions so that they can be modularly operating without constant crosstalk, because things just bind to each other. Does that make sense?
Yeah, it makes a lot of sense. Thank you very much. Super fascinating topic.
I think Naama raised her hand, but
I think so. Maybe Naima raised the hand. Naama?
Yeah, I did.
Yeah,
So,
go ahead.
Quick question, maybe I missed it while you were talking. Could you say a couple of sentences about what the experiment was? You showed some preliminary experimental results, right?
Yes. So those are facts. So I think so.
This is an experiment on cells, or this is in vitro?
No, it's in cells. So it's in cells,
okay.
And it's so there is this technique of polytransfection that Ron Wise developed, where you can you know, probe, like basically you transfect multiple plasmids at the same time. And there's ways to actually control the ratio very precisely. So that's what they do, transfecting plasmids. I believe these are mammalian cells. And then you do FACS to take statistics of single cells.
Okay.
Although I have to say, you know, I showed these data because I think they're really exciting, but they were not taken in my lab. They were taken in Ron's lab.
Thank you.
Thanks. Any other question?
I have another quick question. Could you could you say something if other function may be implemented in similar controllers? So do you think that there can be cells that perform other types of control? I mean,
oh, you mean having cells that have
doing that comparison?
So you mean certain cells are in charge of controlling other cells?
Yeah.
I definitely that's a big question in synthetic biology and even in systems biology. Like think of like microbial populations, you know, that there's a lot of studies now about the microbiome. These are all competing populations of bacteria. I mean, sometimes they cooperate, sometimes they compete, but you can, like when I think of that, I think, well, some of them must be working as kind of a control system for the other cells. So in that case, it's obviously going to be a very complex problem. You know, how do you figure out which population takes over other populations, or how do you maintain a particular ratio between different bacterial species so that collectively they achieve a particular function and they maintain the overall system stable? So there's several research groups that are asking precisely that type of question. That's not my research, but I think it's sort of relates to what you were asking.
Okay, thank you. Thank you.
Any other question?
I have a curiosity, if I may ask.
Sure.
Why do you choose the recombinase-based system for the implementation of the sequestration mechanism instead of like a sigma anti-sigma pair or other mechanisms of sequestration?
That's a good point. Well, one, there's two answers to that. The first one is that recombinases are kind of like a new tool, new in the toolkit of synthetic biology. I mean, they're not totally new. Like they've been around for maybe 5-6 years and people have started using them. So I think there was a curiosity there about trying to get useful behaviors out of recombination. And there's like several groups now. You're actually using recombinases to build logic circuits with memory, because as you invert DNA, you have the ability to record events, right? So one was curiosity, exploration, and the other is that that's sigma anti-sigma factors have been used already to implement molecular sequestration controllers, and so it's in that sense. They have already been shown to work under certain assumptions, usually of low dilution, low degradation. And they work in cells that are slow at dividing, they work. Does that answer your question?
Yes, completely. Thank you very much.
Okay, thanks. Any other question? So there are no more questions. Maybe just maybe an occasion for reflecting a bit on the two approaches, and maybe you two, Naama and Elisa, can have an exchange on this. One thing that impressed me and made me reflecting a bit is the difference of approach in problems that are very similar. So from Naama point of view, more about the statistical inquiry on the data set that she has. But what I saw is that also she implements a model for the signal, the exponential. I don't know if you use it then for reconstructing a mixed discrete or discrete continuous time model or something like that. And on the other hand, the model by Elisa that is more control oriented in the time domain, more framed in classical control theory. And then maybe some conclusions from some debate between the two of you for an exchange of reflections and thought about how these two visions can reconciliate and maybe can be useful one to another towards control, in my opinion. So basically, maybe both of you want to have then the ultimate goal of control.
Should I, Naama, do you want to start?
Elisa. Yes, start.
The way I look at it is the model you use really depends on the question you're asking. So if you're asking about behavior of individual cells, obviously like a stochastic approach is more appropriate, like a point process model is more appropriate. Like our approach was looking at, averaged population level responses, although we did have to, we did look at stochastic simulations as well, not as thoroughly and elegantly as Naama did. We simply took the Gillespie algorithm, brute force, and examined the system behavior and log copy numbers. But obviously, I think the two approaches generally agree once you converge to the same, playing field. When a stochastic approach, you increase the number of cells, she could use a deterministic model and say, for me, if I were to increase the copy number, then I would have to use a stochastic model, but they would ultimately give consistent answers if they put on the same playing field.
Naama, do you want to add
So
something?
there's one more different aspect that we can compare the two approaches other than deterministic or stochastic. And that is the question, how many variables are simultaneously under control? So one of the motivations, I said this briefly, for our work that I presented here, is that when we constructed models, doesn't matter if they're discrete or continuous, if you try to put many variables simultaneously under control, then you run into problems. It's easy to build like a PID or something like that. If you know exactly what is your output that you want to control, and that is very well defined. But if you need to simultaneously control several variables that are dynamically coupled, that's not so easy actually to construct a simple circuit that does that. At least this was our opinion in the models that we studied for cell division. So I think that's also one difference in the approaches. And of course, we know a lot more is known in control theory for single variables, but maybe also many variable control can be used to understand. So gene expression is basically controlled not on its own, but it's controlled in conjunction with cell division control and many other proteins that are all talking to one another. I think in some sense that's a more complex problem, but maybe also methods of multivariable control can be useful here.
Yeah, that's a very good point. And what one should add is also that the key assumption that control theorists always make is, okay, I have a process that is uncertain, but I can build a controller that I know really well, that I understand really well. Now, typically they're built with different parts, right? So the controller is typically built with transistors. It's a computer, so you have very tight, very good control over that. But if you want to build a molecular controller, in the end, it might end up having the same problems as the process, right? So you have to be careful about that. So you need to remember that the price you pay is, okay, I know I have a very uncertain process I want to control, and I'm going to use a set of molecules that I have tight control over, but you should keep in mind, well, do I really have tight control over them? There might be issues even with the controller you're building. The molecular controller itself might have variability and perturbations.
Yeah, I think that these two approaches are very complementary in this kind of having them side by side had raised a lot of points for thinking and reflecting on that. So if there are no other questions or any other open point or anything else that you want to discuss, I would pass to thanking again Naama and Elisa for being with us with a virtual round of applause. And I'm very happy that we succeeded for the second year in organizing these events. And I really hope that anyway, this is going to be the last year that we meet remotely. And then from next year, we can meet in person, or even this year, of course, in other occasions, in conferences worldwide or somewhere. We really want to have a bit of human contact. So thanks very much, very much to Naama and Elisa. As a little sign of gratitude, we'd like to offer free membership to the Italian Society on Chaos and Complexity for the next period. I remember, I don't know, Mattia, it's for 4 years, I think, right? Or something like that. Or it's,
yeah, you are right.
Yeah.
It's 4 years.
So for the next 4 years, so will be our associates. This of course doesn't involve entice any obligations, but just as a sign that you are part of the family. And then as a last thing, I would like to announce the next seminar. Next event will be the 5th of November. It's a Friday, 4 p.m. Italian time or Central European time. We will have Noemi Schmidt and Davide Radi that will talk about dynamics of financial markets. So we had a great time. Thank you very much again for being with us and with the Italian Society for Chaos and Complexity. And see you at next event. Thank you.
Thank you.
Thank you again. Thank
Bye-bye.
you, Elisa. Thank you, Naama.